So today I'll talk about a project that we plan to carry out with the team at now that they're already right, and reach in, and whomever the team is here too. So I was just going to explain what the plan is. The main question, I'll start over here. The main question is this: in the glomerulus of SLE patients, we know the immune complex deposition occurs, and we know that can cause an inflammation. The immune complex, maybe the FC region on the immunoglobulin, or could be the that's probably it. But maybe other things occur like cellular uptake of apoptotic cell debris by macrophages, like function of the mesangium. Those two things, certain things can occur, but this occurs in the kidney. And of course, there's clearance mechanisms, that they could clear too, because we all have this, but in lupus patients, there's a clearance problem. But that's kind of a secondary thing. But the thing is, the immune complex consisting of an antigen, autoantigen, and autoantibody, it has been related to disease. But the question is, it's not clear for this disease what the autoantigen is. Mostly people think of DNA, anti-DNA complex, but it's known that immune complexes of other kind can occur. And so we have observed that Smith antigen, SM, is highly correlated, our anti-Smith antibody. You just measure it in sero patients. It's highly correlated with renal disease, and it's highly correlated with our B cell phenotype, which is high endogenous interferon beta and low IL-4 receptor, those two phenotypes. So, and then that is associated with renal disease, that's associated with anti-SM. And so we have all these pieces, all the way from the beginning, transitional stage B cell development, which may be abnormal, all the way to the end where you make anti-SM antibody and it affects the kidney disease. So what we want to do is fill in the middle and relate each of these steps. So and I can show you how we plan to do this. But here's the final result: is you get that we think SM, and this SM is shown over here. It's this kind of a donut component that binds RNA and processes RNA and makes RNA viable. And it has several components, but the components that are autoantigenic components in lupus are called D and D prime and B and B prime. This may not be exactly right, but it's something like this. That SMD and SMB are the ones that make autoantibodies too. And I'll get into why that occurs in just a second. But so that's why I call this SMD. It may be bound to an autoantibody in lupus, and that goes to the kidney and through the capillary, gets caught in either in the mesangium, or possibly this is a capillary going through here. This is a lumen of a capillary, or it could go through the basement membrane and go to the pseudo podocyte. Could be bound up here and cause inflammation here. So one of the questions is, where is SM antigen? Is it in the podocyte, or is it in the mesangium, and both? And the second question, where is anti-SM? Is it bound to SM it's bound in circulation because we can see the immune complexes, but is it bound in the mesangium, or are they both together here, or does the anti-SM bind to the anti-SM ESN, anti-SM antibody? Does it bind to SMD or B here? So that's kind of the question. How does this end occur? Now, at the beginning, we kind of know a lot about that. What we did was, here's Smith antigen, like I said, the donut-shaped structure. And we have purchased this antigen, the whole antigen. It's purified. Actually, it's purified, I believe, using human serum, using anti-human serum by this company. And they sell us really pure SM, Smith antigen, the whole thing. And we did Western blots, and when we do Western blots of this antigen, and we probe it with serum from lupus patients, we can see the 25 kV band, the 50 kV band, some other bands here too. And they show us a picture of this in the catalogs. We bought the Smith antigen that looks best. You know, has all the bands, especially this lower band. Is it 25 is the lowest or 12?
16 and 28.
Yeah, that's what I was thinking. There's even a lower one down here. I think this is D down here, 16. Something like that. And maybe even 25 is kind of high. They're small. They're kind of small. Each of these components is relatively small. But, so we have this. We have SN. We have anti-SN. And I just say a little bit on the side here. This is not something we want to work on too much, but let's say SMD2. Maybe this one goes down here. SMD2. D3. SMD3 can become antigenic if it's modified. And the modification that occurs on that is been studied. It's called symmetric dimethylarginine, SMDA modification. So what that means, here is a close-up of D3, right? D3. And what you see is arginines every now and then. There's amino acids in between. But it turns out that if they're symmetrically dimethylated, so this CH3 group is coupled to an arginine, this is so dimethyl symmetric means both. This makes SN as a peptide more antigenic. People have looked at this. And there was a paper that showed that even if you have two of these, and they're the right, somebody made this peptide, made SN, this is SND3, you know, SN, it's SND3. That's a component, and it's known which amino acid sequence it is. If you have two symmetric dimethylarginines, I think it was like 7 or 8 amino acids apart, that's really antigenic. So that's why I drew here, you know, things in between. Now, SND3, this region and the protein in general, is semi-positively charged. I put that down here. So they tend to be positively charged. DNA is negatively charged, and the anti-DNA antibodies are somewhat positively charged. But Smith antigen is positively charged, and we propose that the antibodies that bind to it are negatively charged. That's just one of many properties. So let's look at this is SN, this is SMD. So we know what the target is. I mean, we have. Well, I'll get into this in just a second. But we know how this works. Everybody has studied this for a long time. And we also know the antibody. The antibody, we have looked at SLE patients, and we looked at ones that are SN positive and SN negative, and we can look now using the using the 10x 5' sequencing component of 10x, which you can get the B regions of heavy and light chain of B cells, so we can get all the antibodies of SN positive patients. We don't know which ones are anti-SN, but we do know that patients that are positive tend to be anti-SN positive and have renal disease. They have an expansion of a particular population of a heavy and light chain region in those B cells that tends to be negatively charged. You know, we could show you that -8, -9, so they tend to be negatively charged. So we think this. So here's what the antibody is. Nobody has actually ever studied the antibody, the anti-SM antibody, right? I mean, nobody knows how to do that. So that's what that's what we're going to do. So this is the anti-SM antibody that binds to SM, and here's the heavy chain, which is red, and the light chain, which doesn't have all the several constant regions, so it's lighter. It has a V region though. And both of these chains can now be isolated using the 5' method of 10x. And what that is, I just mentioned real quickly. There's V, D, J, and C. That's a heavy chain. And the light chain is V, this is heavy, and J and C. C is lambda or another chromosome kappa. And it's on another chromosome. So this is on 12, I think this is on 6, and this is on 11 chromosome.
22 maybe?
22, yeah. Something like that. So there's, and what happens is, the first thing that happens when you rearrange an immunoglobulin at a very early stage is even in bone marrow, the pro-B cell, pro-pre-B cell.
V combines with J. There's about 20 active V's, there's about 6 J's, and these recombine, probably in a stochastic random way. And then later on, its DJ join binds to the V region. That's it. That's the heavy chain. That moves this promoter down here at the end of the heavy chain, very close to this enhancer. There's an enhancer in here. I believe it's in here. T-cell receptor has down here. But I think immunoglobulin enhancer is here. And then you can transcribe that. And that enables a light chain, either be lambda or kappa to rearrange. And that's simpler because it just V joined to J, and then this little gap stays open. And then, but again, the promoter is moved closer to an enhancer. But actually, this all together is enabled. This is first, this is second. This stays pretty much fixed. This can switch to that. Different things can happen at that level. But we know from our sequencing data, from the 10x sequencing, we actually can target this and we can sequence and identify the heavy and light chain for like 5,000 B cells in peripheral blood. So it's a huge number, a huge amount of immunoglobulin information. And we're actually pioneering a program that can sort of do bioinformatics analysis and look at that. We just looked at one, a plasma blast. We did a heavy and light chain. There were just two prominent, just drawing it. There's two prominent heavy chains and two prominent, this is a big thing, two prominent V heavy and two prominent V light chains that were prominent in the plasma blast that's over here of an SLE patient. So it may not be hugely complicated. I mean, it probably is a little complicated, but it's not like, it's really restricts down the repertoire when you get to the plasma blast stage. And, so we're working on that to try to figure out what they are bioinformatically. But anyway, we're going to know it. We can get this. We can clone this once we have the sequence. We send it to some place called Twist. This company in Texas. They will take our sequence and they will make an antibody. So we can, if we find an antibody that we think is anti-SM, we can produce the antibody, the actual antibody, and then we can test it, like on a Western blot. We can see if this antibody actually does recognize SM and to what is the avidity and so forth. So this has all been worked out sort of in the past, but we're still working on it. Okay, so that's the antibody. Now, here's the interesting thing that we can do. Just recently, is because we use the 10x sequencing to get the heavy and light chain, it also gives us the entire transcriptome of that same B cell, about 30,000 genes. And using that method, we already show, and I guess that's a little bit over here, we already show that these pathogenic B cells tend to have what we call interferon IRGs, interferon response genes, positive like MX1, a lot of those, and then if you plot IL-4 receptor as a gene or a protein, it tends to be low. So they're all sort of in this quadrant right here. A lot of the B cells that are pathogenic are lupus. They're MX1 positive, IRG positive, and IL-4 receptor negative. Normal people are down here. This is normal. And it's a simple, I know it says, well, it can't be that simple, but it might be that simple. And these are the ones that are pathogenic, and those are the ones that make these anti-SM autoantibodies. So that's the transcriptomics component, and this is the immunoglobulin component of the B cell. We know everything. And using the 10x, we can also identify what stage the B cell develops. For example, I just drew in here the phenotype of a transitional T1 B cell. Transitional stage B cell is the first B cell that we can measure in peripheral bloods. There's, if you do an experiment, you get about 1,000 of those. Or we can sort for them. We've done that. We can take that, for instance, transitional. But the question is, when does this pathogenic autoantibody get made? Like I said, well, I didn't say it yet, but after you make this rearrangement of having a light chain, then other thing, as the B cell develops, it has to be selected. And normally an autoreactive B cell that recognizes anti-SM would be not positively selected, or it would be eliminated during development because it would be autoreactive. That's how we get rid of those autoreactive B cells. So at least it wouldn't develop to a pathogenic stage. And again, we will know, because we'll know the antibody that's produced by a B cell at every stage of development. We will know, we will be able to identify the sequence and identify if it's pathogenic. We will have an antibody, I didn't get that, I don't think. We will have an anti-SM antibody, we'll talk about that in a second, that can recognize every type of anti-SM by the idiotype. That's this one here, I was going to say. And of course, we know what SM looks like. We have a lot of anti-SM antibodies because we purify those on the column. So we have all the tools in place. Now, so here's the plan. We can sort, or we can identify by the transcriptomics and kinetics what's the transitional B cell. Let me just show you the phenotype here. It's a transitional B cell. It's not memory, so it's CD27 negative. It's early stage IgB positive. And in humans, transitional B cells are CD24 positive, CD38 positive. So it's in a little upper corner that we can date on. And some people use CD10 positive. So this is T1, or CD10 positive. So these five markers, which we can identify both in flow cytometry downstairs or by transcriptomics, can tell us whether it's a transitional B cell. Of course, then we know naive B cells, activated naive. Activated naive is the one that has Tbat, for example. Plasmablast is CD138,
Yeah.
right?
Plasma cell number is CD138, but
138.
I'm not sure plasmablast, but CD138 is it.
Right, and plasmablast too. And transcriptomically, what do we?
CD38, 27, double positive.
Right, CD38, CD27 positive. These are genes, so I should put it in the tab.
That's a protein.
Oh, protein. But what about the genetic gene?
Gene, I think it's a link 1.
Oh, okay, you throw in the one. So, but I guess what I'm saying is, by flow cytometry or by transcriptomics, we can look and identify a state, the B cells at every stage of development, from naive, from transitional to plasmablast. So, so that's that B cell. And we can tell, using this method, what the immunoglobulin is they express. So what's missing? The missing component that nobody has, but we're making it, is we need an antibody that can bind to these anti-SM. We don't know which ones are really anti-SM. So what we're doing is with Tom Joe at a lab down the street, he made a column of SM. We bought this SM from a company. Remember what it was? Company? Immunocreate. Okay. This company, and we looked at a lot of companies. Immunovision. We looked at a lot of companies and a lot of SMs. There were a lot of people and companies sell this. But we got the best. You know about it because it's probably used in pathology. And a test is what you coat the plates with, to see if somebody's anti-SM positive or not. So we looked at a lot of SM, and Immunovision, I think it was had the best anti-SM. We looked at the Western blots of how that looked using sera of SLE patients and normal. And I'm so negative, we did our own Western blots, and I'm just 100% blank. I have zero SM, anti-SM. Okay. So very quickly, what we did, we took this SM, the best SM, and we made an SM column. Then we took SLE sera, and we screened it for who's SM positive, so we had good sera. We have 150 patients, but we took the best sera, which we saved, and we put it over the column. We made we purified anti-SM antibody, real antibody. We know it's very good. And then we injected that into a mouse and made hybridomas. And the identified hybridoma shown here, this is a hybridoma, a monoclonal antibody, that now recognizes it's an anti-SM idiotype. That's not available anywhere. Yeah, so, but we have it. And it recognizes a key component that's only expressed by anti-SM antibodies, not by any other one, because we did the positive and negative screening. And the idea was, I was just asking Sherman, is to barcode that with a 16 base pair tab. It's a sequence, a unique sequence, that when we do that, that when we take the B, you know, what the plan is B cells that express this antibody, B cells that express this antibody can be identified as being anti-SM because we have this anti-id to them. We can put those all into the 10X and we can look at those, which antibodies have this, and they'll come out. We'll see them. We can pull those, that we can identify them bioinformatically. We can look at their having a light chain. We can make the antibody with twist. So, and then we can use transcriptomics to go back and look at when did this B cell make anti-SM? Did it make it at the early transitional stage and so forth, all the way through to the plasma vessel? Are they enriched? Does this kind of antibody get enriched as in SLA patients compared to normal? So the idea is, the idea for the project is to take a lot of our lupus patients, I guess as many as possible, and try to study the kidney disease, relate their anti-SM and their disease to kidney disease.
Do you have a selection criteria for lupus patient, or is just in general? Because not all patients are positive for anti-Smith antibody. So do you want, like, are you gonna include only those patients who clinically have anti-Smith antibody, or just in general?
I think at first, probably all kidney disease, anybody with kidney disease, because if they're not
The
anti-SM.
control and we can put like the control without.
Yeah, because it'd be interesting, because it's not, we want to look at the kidney, and if they have kidney disease, but they're SM negative, that'll be interesting. I suspect, we know that those people will not have this phenotype. They won't have a lot of B cells here. Of course, they won't make SM antibody, and they have kidney disease, probably they have anti-DNA or something, DNA immune complex. Maybe they'll have DNA, anti-DNA in the mesangium, preferential to the basic membrane, basic membrane or the podocyte. The reason why I say the podocyte, because there's one paper, remember, what's his name? He's on all the Appel. Remember this Appel, the kidney guy?
Mm-hmm.
He writes all the papers about the kidney and immune complexes. What's his? Appel, A-P-P-E-L.
Yeah.
He seems like he's always writing these papers. For years, years ago, I saw papers by this guy. And he had a table once, I think he showed it to you, but we have it here, that he looked at a few patients, not very many, and he looked at what kind of disease they had, and he had mesangial-proliferative disease,
Mm-hmm.
and they had anti-DNA, but no SM. He looked at membranous, type 5 membranous.
Mm-hmm.
They had anti-SM and non-anti-DNA. So, of course, that's a small study, and maybe it's not perfect like that. And we talked to Wynn, of course, most kidney people, most SLE patients have both mesangial-proliferative of some degree and membranous. It's probably not many, just purely membranous. But, of course, it could be they have antibodies to DNA, anti-DNA complexes. They go to the mesangium, and I think charge has something to do with it. Remember I said they're opposite charges? Because you were explaining to us, you were explaining this little barrier, if you blow it out, is very complicated. It's got glycoproteins, here. I'm not doing a good job. This is the lumen, this is the capillary. And there's a podocyte here, I think, something like that. And these glycoproteins that are expressed on this part of the lumen, I'm not exactly sure where, but they're glycoproteins, are highly charged. And these charges, remember I said this is positively charged, this thing is positive, SM is positive. The antibody is negatively charged, and I believe that these glycoproteins confer a positive charge. Is that right, Bill? Or is it?
basement membrane is heparin sulfate rich, which is negatively charged. And then in a normal individual that is required because we need to prevent albuminuria. And albumin is negatively charged. So it provides a charge barrier in a normal individual. I'm not sure how glycocalyx is. Maybe it's positively charged, you're right.
Glycocalyx in which one?
So basement membrane in itself. So filtration barrier is fenestrated endothelium. Then basement membrane in itself, glomerular basement membrane, and the foot processes with the slit diaphragm. So these are three components of filtration, glomerular filtration, that provide glomerular filtration.
Okay.
So of these, the basement membrane contains heparin sulfate.
It's basement membrane.
Yeah, and that gives it a negative charge, and that prevents albumin from passing through, because the fenestrated endothelials, each fenestration is bigger than the size of albumin.
Right.
So if there is no negative charge, albumin will go through the pores in the endothelial cell cytoplasm, and it will spill. People will be spilling a lot of albumin normally if there are no negative charges in the basement membrane.
Right, so this is negative.
Yeah, and that is
in the
negative.
basement membrane.
Yeah, but
Which-
glycocalyx, I don't know. I'm sorry. Maybe I have to read about glycocalyx.
Well, the glycocalyx, somebody did an experiment just sort of semi-recently and wrote about it. But in any event, he got rid of the glycocalyx with some sort of
I'm sure that has resulted in proteinuria too.
Right, yeah.
Yeah.
He didn't show the, it's funny, he didn't, there's all these things they didn't show. What he showed was there was an increased cellular infiltrating. I don't think it was a very good study, because he didn't show the control without the glycolytic enzyme treatment. And then he had a prevention method and so forth. But, so the glycocalyx is one thing, and I don't know whether that's abnormal or not in lupus, but that's almost another study. We can study carbohydrates. We did it before, but we couldn't do so much. There's the basement membrane, which is negative, right? So that's very important to know. I don't know what charge it is. Maybe it's negative, but you do wonder if let's say everything's negative because albumin doesn't get through. So everything here is negative. Let's say, this sort of negative. Albumin doesn't go through, and the other thing that won't go through are negatively charged antibodies. If an antibody has an overall negative charge here or here, it might not go through very easily. So, but this, at least SM, is positively charged, right? Very positive, okay. So maybe that's an argument for SM is somehow being metabolized in hepatic cell debris, that's how little bits of SM component, SMD. It's very small, by the way.
If it is positively charged, it should go through the basement membrane.
Right.
Because it's positively charged, and it goes through and that makes sense because you said that in membranous it's more Smith antigen deposit, right?
Right.
So for subepithelial deposit, these antibodies they have to go through the basement membrane to lead the antigen.
Right. So it may be as
Makes
simple
more
as
sense.
a positively charged antigen, which is small. It's what is it, 8 or 16 kD.
Oh, I'm sorry. That's the antigen which is positive, and antibody is negatively charged. Oh,
Yeah,
okay.
the
Sorry,
antibody-
that makes sense.
Well, we don't. Yeah, the antibody. This is positive, but the antibody might be negative. These are the ones we looked at, but we don't know. So, but maybe this goes through, and I don't know how the antibodies get through it then. Or maybe it goes through as a complex. Maybe the overall complex with antigen and antibodies. But this is all things we can look at. If we have, once we look, first I was thinking of looking at as many kidney sections as you have, having Fatima review those, and then kind of identifying interesting cases, and seeing if they're membranous or, type 5, I guess, or 3 and 4, which is membranous, mesangial proliferative, or both, or neither, whatever, or neither. I'm not sure, but I guess neither doesn't, probably both. Since they're kidney problems, they might have one or the other or both. And then we have the data on those patients. We'll give you the list of patients. We do have the data. We have whether they're Smith positive, anti-DNA positive. We know a lot about the patients. We know about their immunology. We have the sera. We have a lot of things on these patients. But what we don't have is detailed information about the kidney, whether they have membranous membranous or membranous proliferate or whatever, you know, or glomerular.
Class 3 or 4, but yeah.
But it class 3 or 4, we don't know. And I know you know, so it's there. So that's one thing. What does a kidney look like? Well, and then we get some that are available. You said it takes after you sign them off. Well, we could do a lot more. Go back.
What kind of paperwork we need to be able to study the kidney? Do we need to get the IRB approval, or how do we?
If it's a human tissue, definitely nobody is gonna
Right.
like issue it. So my thing is, that what type of tissue are you expecting from pathology then? And have you?
We didn't.
That's,
We didn't do any, but just.
I know kidney, but
I mean, right. The first thing is just the kidney, and maybe the light or EM that you did, the results, or just
okay,
or
just
even.
the results will be fine. I mean,
Okay.
but if you need a real tissue like a remnant, that we won't be able to give you for
Right.
research.
Right.
That's diagnostic tissue. And if you are in touch with Dr. Whom you have a meeting with just now, Dr.
In Chatham?
Chatham, yes. So they can actually take consent of the patient and do an extra core for the research purpose
Mm.
if you want a real tissue.
Mm.
But if as long as the report is you want to report findings, we are more than happy to provide you. But even then, that it requires, I think, some kind of approval.
Okay,
If not
and
real
we-
tissue, there is a category for IRB approval. I mean, it will they just want to know that they will be properly de-identified, the information, patient information. That's the only thing. But if there's no tissue,
But can I just say one thing? One thing you mentioned before was the possibility of, you said you have frozen sections that are available, like on a slide.
yes, frozen section we can
Clean it.
provide, but that will be an IR.
Right, and we get IRB.
Yeah.
I think that's really what I want, because maybe we may get around to an extra core for later use, what I mean, for research purposes. But that's kind of extra.
Yeah, that's a favor lab can do, that we can spare frozen tissue.
Right, but we'd have to get
Right.
IRB approval and so forth. So we don't have that, but to just get a slide, a frozen section, that the case has been signed out and everything's done.
Yeah. The slide for what?
For frozen section, for staining, for example, staining, we run a stain, we have
So,
an anti-SM antibody, stain that slide and see.
Are you doing IFs, immunofluorescence on these?
Yeah.
Right.
Okay. Okay. So one thing is so that you need IRB approval, and that you need patient consent. That their tissue, we can't just because that's what I'm saying. Dr. Winchatham, you have to bring him in a loop. That if he has to tell patient that his tissue is going to be, if it's okay, they can use that in the research.
Right.
So we need, yes, the consent and because the lab is not going to release without having a consent.
So we do have, so if a patient comes back and we reconsent them with a new consent to study their kidney slot.
Yes, yes.
This is not to do an exobiopsy, just,
Dr. Mons, it's not possible for us to give you already stained section. We will give you unstained slides.
yes, right, that's
Yeah.
what, that's what we like.
Yes, unstained slides. We will give you unstained, but with the patient consent.
Wait a minute, okay, so
Do you guys
that's,
have amnesty slides?
No, we'll cut for you.
Oh, okay.
We don't
You
cut.
have like extra, like.
Whatever tissue will remain, we'll cut unstained for you.
okay,
For
well, that's,
immunofluorescence.
I mean, I bet that's about what we want.
We don't
If we-
cut necessary. So we do 3 immunoglobulins, 2 complements, and 1 kappa and lambda. So they that makes 7 and 1 negative. 8 slides, 8, right? A normal immunofluorescence.
Right, oh, that's what you do.
But yes, that's
But
what
you
we
were gonna
always.
that's
store it
a
for
lot.
how many years?
Huh?
You will store it for how many years?
The slides just 1 month.
So like we were not gonna able to access like any slide like in the past 3 months or in the last year.
No, we save the tissue
Yeah, okay,
forever.
forever, okay, got it.
Oh, she
Especially
means
for-
she'll cut a new one.
Yeah.
Right.
Yeah, we will cut.
From
it.
that, to find the
From
tissue.
the-
If we
So-
get consent, she can locate
You
the
cannot
tissue.
use the user which have already
Yeah.
been
That's
used for IgG, IgA, IgM, right? They already have anti-zero on
Yeah,
them.
yes.
So you need new
Yeah.
unstained sections,
But
right?
what would be interesting, okay, we got this list of patients, maybe 100 long, and then you get the path report, you say the path report, and you got all this stain already, heavy chain, light chain, and
Yes.
complement, and you know whether, what class it is, all these things, all the
Yes,
histology.
yes.
You get that report, and then when you pick out some that look like, okay, membranous or whatever, pick out a sample, 10, 10, 10, say 30 interesting ones, or start with 5, 5, and 5, membranous, stage 2, 3 or 4 or 5 or both or none, and you sort of, I don't think you have a none, but then you would, and then we would get those 5 or 10, and then we ask when, are you still see these patients, right? And then you say, can we reconsent these patients? And you go, okay, well, she's coming in.
So, Normans, here is the
Yeah.
thing. I think if it's just for now, it's just the data, like computer data you're using, like per
Yes,
post.
computer good.
So I don't think so. You need patient consent for that.
No. But what I mean is that to
But
get
if
your
you're using the tissue, then I think it's ethically.
right.
should get
I mean, the
consent.
first step is to
But
get.
on the other hand, your selected patient will know your blood profile.
Oh, Fred, yes.
So we kind of, there's some patients.
In about 10 minutes, I think it is.
So in that case, maybe the right selected patient, we know their blood profile, the immune profile.
So maybe what you don't need IRB or you need later.
So with those if we already know who we want to see
Yeah.
their report, in that case,
That's what I'm saying. If you're just limiting to the computer generated data like pathology report, I don't think so. You need patient's consent to look at the report. It will be just you have to de-identify the. That's what my understanding is. Some of the IRB, which I have, the courses I have done for IRB, but you better check from them that this
Yeah.
be like clear.
right.
Make it then clear what you are gonna use, and they will tell exactly what do you need to tell patient
Yeah,
and what.
that's right.
Okay.
Yeah.
But we do have a connection with IRB, a very good connection.
Yeah.
Remember-
I mean, IRB was definitely.
CCTS, there are 2 people there who are working. And what's her name? Hina, yeah, mine too, good. Not Homer, but Hina, some person.
That's right.
Hina, yeah, right. So there's somebody over there who's very good, and we work with her, and she'll modify the IRB, because we gotta do that anyway. But in addition, she can tell us if what subject data we can access. So that'd be one meeting with Win and Hina. And
When,
Hina.
is this a prospective study or one's a retrospective? Are gonna explore previous patients?
Yeah, retrospective, because we can start a prospective one, but that'll.
Where are you gonna get patients list?
Where to get the patients list?
Yeah. You have already listed.
The patient list, I think
You already have it?
Have some for our own
And
studies, we
so
have-
all of them are biopsied here at UAB?
Not biopsy.
Not all, but-
BNC.
Oh,
BNC.
okay.
Oh,
Do
we
you
have
know how
BNC.
many have been biopsied?
We don't know.
Oh.
That we have no idea.
No, we know that kidney, when it told us they have kidney renal disease or not, but I don't know what the histology showed. So we'd start with the ones that he says these have renal disease. Did he?
That's what I'm not asking the hormones. Do you know how many they get biopsied and